Good morning, students. Today we are going to discuss. Today we are going to discuss about the cell wall. Discussing about the cell wall. What is the cell wall? We are going to discuss about the cell wall. This is a unique feature seen in plants. Cell. What type of unique feature? Unique means one identical feature, one special feature. What we are going to see in the plants, that is called as the cell wall. While the cell membrane acts as the outer layer in an animal cell, in a plant cell there is an extra layer. There is an additional layer in the plant cell, mainly of cellulose outside the cell membrane, which is known as cell wall. The outermost layer of the cell membrane, as I told yesterday, that is the outermost layer of the cell membrane. Cell membrane. Cell membrane outermost layer is called as the cell wall. This is considered to be one of the major difference between the plant and an animal. It is considered as one of the major differences in plants. So, in plants as well as animals, there are difference. So, there is various difference between the plants as well as animals. In plants and animals, what we are observing, so in the last class we observed the plant cell. They are circular, regular, or some are irregular in shape. They are highly irregular in shape. So, this is the arrangement. All the cells are not same, all the cells are not regular, all the cells are not irregular. They are arranged in gigantical manner in each different plants as well as animals. Here we are seeing the outermost layer of the cell membrane is called as a cell wall. This is considered to be one of the major differences between the plants and animal cell. There is a lot of differences between the plant cell as well as the animal cell. We cannot coincide each and every cell. All the cells are not similar, like how I have drawn here. So how I have drawn here, there are some up level to upside, some are horizontal, some are like this, some are vertical, some are broken. All these type of cells we are observing in the cell. That is all they are not similar to the animal cell as well as the plant cell. Then the cell wall is a tough but flexible porous layer. As I said, they are tough. How they are? They are tough, very tough and porous. Meaning for tough. Tough is very hard. They are very elastic. We can say other word elastic. We can say the other word, they are tough or elastic in nature. Then they are porous. Porous means here they are permeable. Porous means they are very permeable. They will allow the required nutrients and other substances as well as they will exit from the other end. They will exit from the other end. Mutual exchange of gases also takes place. Exchange of gases also takes place. So the arrows what I have given here to represent that whatever the things are there required for the plants, these plants are collected inside. These materials are collected inside. So after collecting inside, the gases are such as carbon dioxide and oxygen is going to be evolved. When it is going to be evolved? When there is a degradation, C6H12O6. Whenever the glucose is going to be destructed, it is converted into carbon dioxide and water vapor. Water vapor. Carbon dioxide and water vapor. They are converted as carbon dioxide and water vapor. This carbon dioxide and oxygen molecule is going to be exchanged. There is going to be mutual exchange. From where this exchange is going to be observed? This exchange is going to be observed from the porous membrane. From this porous membrane, the gaseous exchange is going to be observed. Not only from this, the tough and elastic membrane, the tough and elastic layer, that is the outermost layer of the cell membrane. Next, continue. Page number 3, we can continue here. The cell wall is a tough but flexible porous layer that tends to different shape and gives protection to the cell. The cell wall is the tough and but flexible. It is tough, as we are saying, it is tough, elastic, but it is very flexible. Flexible means what? They can easily enter, they easily allow the required nutrients, minerals and other substances into the cell. Though it is tough, but it is very porous nature. After that, earlier it was believed to be inactive. Earlier it is we consider that before the scientists had observed, we are seeing that they are inactive in nature. But now it is considered to be one of the most significant part of the cell. But now, after the scientists have observed what is the importance of the cell membrane, then they come to know that no, it is not a waste material. It is very valuable material. It is very porous material, very important material, important constituent of the cell. That is the outermost layer of the cell wall. Important constituent. So how we can say important constituents? Because it is a porous in nature. It is permeable. It will allow the nutrients and exchange of gases such as carbon dioxide and water vapor. So all these things are happening because of this membrane, this porous layer has got such much importance. This much importance has observed because they are porous in nature. It will allow the mineral nutrients and give the exchange of the carbon dioxide and oxygen as well as the water vapor. Now we can continue, but now it is considered to be one of the most significant part. Next, continuously exchange information with the cell during growth and development. So by doing this, they will start their growth. Next, what are the functions of the cell wall in the plant cell? It exerts an inward wall pressure to resist the outside pressure. To resist means to avoid the outside pressure. It exists an inward wall pressure. Next, exerted by the cell space, cell sap. Hence, the plant cell can withstand much greater chances of surrounding medium than the animal cell. So, because of their, what is they are saying here in the cell wall? We can observe here, whenever the external pressures are observing from the outside pressure, they themselves will create the internal pressure. They themselves will create the internal pressure within the cells. Within the cells, they will create the internal pressure. Once they create the internal pressure, the external pressure is going to be avoided. This mechanism is happened only in the plants. They are saying that this type of principle, this type of mechanism is happened only in the plants. But animals, the cell wall mechanism is again different. There is very changes in the animals. When we go there, then we can discuss there how the cell wall is going to be functioning there. But with respect to the plant cell, we can observe the mechanism of plant cell, how to avoid the external pressure. We can say here, they are creating their own internal pressure within the cells. Within the cells, they are creating their own internal pressure. Thereby, what happened? By doing this thing, the external pressure can be avoided. So once we can recall what is the cell wall, we can say here, it is a unique feature of the plant. Cell wall means it is a unique feature, having a specialized feature in each plant. And we can say that the cells, whatever present in the cell, they are very different from each and every plant. Each and every leaves, branches, stems from one plant to other plant, they are very peculiar in shape, peculiar in size, peculiar in structure, peculiar in color, peculiar in their growth, peculiar in their behavior. Everything, each and every aspect is totally different in the plants as we are observing. So when we go for outside, example I am saying now, when we go to outside, when nearby from your house or from the school, outside of the school, when we observe the locality in the nearby area, we cannot find all the same plants in each area. We can find there is a lot of differences, lot of variations we can observe, lot of changes we can observe in each and every area. What are the changes we are observing? There is no similar shape of the plant, there is no similar shape of the leaves, no similar shape of the branches, no similar shape of the roots, no similar shape height of the plant. Each and every plant height is varying. They will change. They are not having the same height. Some plants will grow up to the height of 10 feet. Some plants will grow up to the height of 20 feet, 30, 40 feet. If forest, if we see, in the forest if we go and observe, there is a lot of changes. So many changes we are going to be observed. So at a very height of we are going to observe the plant. Not all the plants are having similar shape and height. Likewise, we are trying to say here that if you go inside the cell, the outermost membrane, outermost layer of the cell is called as the cell membrane. The cell wall is present just behind the cell membrane. They are saying that the cell wall is present just behind cell wall. The cell wall is present just behind the cell membrane. So just the inner side, outermost layer, we can say here the cell wall. So inside the cell wall, so many cells are there. This cell wall act as a protective layer, act as a protection. Protection from what? From how many things they are going to protect? From which things they are going to protect? They are going to protect from the harm, HR. We can say they are going to protect from the harm. Harm such as harm from destruction, harm or destruction from the outermost layer. From the outermost layer, we are going to protect from the plant. Distinction from that is external pressure. External pressure. P-R-E-S-S-U-R-E. External pressure. So it will protect from the external pressure. So that what happens? You can observe here the inner layer. The inside arrow mark indicating that from the cell wall they are creating inside pressure, internal pressure. This arrow mark we can say they are creating the pressure. They are creating pressure. What pressure they are creating? They are creating the inside pressure. So by doing the inside pressure, what happens? The external environment pressure, whatever the pressure of observing, that can be minimized. This is the internal pressure, and here you can write external. The external pressure, we can write the external pressure. But when the external pressure we are observing in the plants, to avoid or to protect from the external pressure, this cell wall is protecting the inside cells so that these cannot go for damage. These cells cannot be damaged. It will protect the entire way. In all the ways they are going to protect to prevent the damage or destruction of the cells. Therefore what happens? These cells are going to be protected. So that is the thing, cell wall. That is the function of the cell wall. So again what they are saying in the cell wall, on the plants, not all the cells are similar. They are different. They are not at all same. They are different from each and every plant. They differ from one plant to another plant. They are not entirely the same. They differ from shrubs. They are different from herbs, from the forest area plants, from the cultivated land plants, grass plants, or paddy field plant, papaya plant, grape plants, onion plant, banana plant, and some other type of plants are there which are not similar. Each inside the plants, if you observe the cell position, they are not the same. Their arrangements are different. Their behavior is different. Their growth is different. Their size is different. Color is different. Everything is different. They are not at all the same. They are having each and every peculiar structure. Peculiar means one having identical importance to each and every plant. Not all the plants are having the similar structure. Not all the plants are having the similar cells. They perform their own unique behavior. They perform their own characteristics. Hence, we can say the cell wall is going to perform a various type of functions. Not only from the exchange of gases, but also will act as a porous for gases exchange. Act as a tough elastic membrane to protect from the external environment. Because as I written here, they have to protect from harm, destruction, as well as external pressure. So to withstand the external pressure, the cell wall act as a protective barrier. The cell wall act as a protective barrier, protective layer, whereby we can prevent the other things. Next, we can go for next one, that is nucleus. We are going to discuss the second part of the discussion, that is called as nucleus. This nucleus can be studied with the help of lab activity. We are going to write an example, that is the aim. What is the aim? Aim is to observe the nucleus under microscope. So to observe the nucleus in cheek cell. So to observe the nucleus under the cheek cell. So we have discussed about the cheek cell. Once again we are going for revision. What is the material? What are the things required? Material required. So material required one means once I will explain you orally, then I will go for explanation. A toothpick or ice cream spoon or spatula, glass slide. So whatever the things we require, that is toothpick, spatula. Another thing what we require, that is a slide made up of glass. Glass slide required. Then cover slip to cover the sample. Cover slip. After that, watch glass. Watch glass. After that, needle. We need the needle. After that, what we need? Needle blotting. B-L-O-T-I-N. Blotting. B-L-O-T-T-I-N. So blotting. Then paper, blotting paper. So this is blotting paper. Then 1% methylene blue. Requirement methylene. 1% of methylene blue. Then saline, normal saline. Normal saline. Then glycerin. Glycerin. And the last and very important thing we require, that is called as the microscope. The microscope. Now listen, these all the things we require. Why these all things are required? To see the nucleus in the cheek cell. Where we are observing the nucleus in the cheek. Cheek cell. To observe the nucleus in the cheek cell. To study the nucleus present in the cheek cell. Now, listen, what are the materials? These are the material here. What is the aim? Aim of the thing is to observe the nucleus in the cheek cell. First one, that is toothpick. We need a toothpick. Spatula. Spatula, I am writing for the rough diagram. That is such as spoon. Spoon name of spatula. That is spoon. One spoon is required. One spoon is required. Then the glass slide. We require the glass slide. Why glass slide required? Because to place the sample. To place the sample on the glass slide, we need a glass slide. To put the sample. Sample మనం తీసుకున్న sample దానిపైన పెట్టడానికి ఒక గ్లాస్ స్లైడ్ అనేది అవసరం. We need a glass slide. After that, what we need? We need a cover slip. So we have taken a slide, we have placed the sample, and we need a cover slip like this. You can observe here. I am writing the parts. This is called as slide, glass slide, and here is the sample. Likewise, same, and here is the cover slip. So I am trying to explain in detail what is the purpose. Why we have to collect all these things? What is the use of these things? One by one I am going to explain you. So glass slide, outermost layer. This is the glass slide. Cover slip, the cover slip, and the sample. We have kept the sample here. Next, needle. Why we require needle? Needle is required to place the cover slip. To place the cover slip. Next, blotting paper. Why we require blotting paper? Blotting paper is very much required to collect the excess of glycerin. Excess of glycerin. So after placing the glycerin on the sample, if it is spreading more, చూడండి ఇక్కడ ఏమైతుందంటే ఎప్పుడైతే కవర్ స్లిప్ మనం వేస్తామో, whenever we place the cover slip with the help of the needle, before this we have to add the glycerin, 1 or 2 drops of glycerin. When we place the glycerin, some glycerin is coming outside like this, glycerin coming outside. To prevent the excess of glycerin, we are using the blotting paper. What we have to do? We have to just touch the corners of the corners of the cover slip. So that what happens? The blotting paper will absorb the glycerin. It will suck the glycerin. Then 1% methylene blue. Why you have to add the methylene blue? This methylene blue acts as a color producing substance. That is the purpose of the methylene blue. After that, we have to add the saline. Saline, purpose of saline is to prevent the shrinking of the cell. It will make the cell to shrink. So that what happens? We can easily observe the nucleus. Then glycerin. So we are adding here the glycerin. Glycerin is added to not to disturb the slide. Not to disturb the slide. After adding the glycerin, the slide is not going to move. It will be remain the same place. Then microscope. You know the microscope to observe. So observe under the microscope to see what is actually the sample contains. The nucleus, what we are going to look now, what we are observing the nucleus, we have to clear image. దానికి ఒక ప్రాపర్ న్యూక్లియస్ ఇమేజ్ మనం కనపడడానికి మనం మైక్రోస్కోప్ we are using the microscope. The resolution is 10x, 22x, 25 and 44. X. What is the meaning for X? It's indicating the resolution power of the microscope. The resolution power of the microscope. We are going to observe under 10x, 28. So normally, if you want to observe the nucleus, we have to observe in the highest resolution, maximum resolution. We have to observe. When we observe in the maximum resolution, there is a more chances of getting the clear image of the nucleus. Clear image of the nucleus. If you want to get the nice picture of the nucleus, we have to observe all these things. Now, I will read the procedure now. Wash your mouth and scrape a little of the internal lining of your cheek inside your mouth with a clean toothpick or spatula or ice cream spoon, with the help of the ice cream spoon. Then place the scrapped material in a watch glass containing a very small quantity of normal saline. So watch glass, I have not written here the purpose of watch glass. This watch glass is available like this way. It is available in this manner. It is available in this manner. Why we are using the watch glass? After collecting the sample, we have to place here. After placing, we have to add the glycerine. We have to add the glycerine. Uniformly, we have to distribute. We have to add with the help of glass rod. We have to spread. Glass rod ద్వారా మనం యూజ్ చేసే వాచ్ గ్లాస్ శాంపుల్ ఎప్పుడైతే చేస్తామో దాన్ని యూనిఫామ్గా మనం స్ప్రెడ్ చేస్తాం. దానికోసం మాత్రమే మనం ఆ గ్లిజరిన్ అనేది యూజ్ చేస్తాం. నెక్స్ట్ వన్. Then place the material on a glass slide. After mixing the sample from in the watch glass, we have to keep it on the slide. Next one. Put a drop of methylene blue. Then we have to add the 1% methylene blue solution for a couple of minutes. And we have to wait for some minutes. Put a drop of glycerine over it. After that, then you have to add the glycerine. Then place the cover slip. Tap the cover slip with the blunt end of needles so as to spread the cells. So to spread the cells, we have to tap with the needle to light kottali. Wipe off the extra strain with a fine cloth or blotting paper. Whatever the excess of liquid coming outside, that we have to tap with the blotting paper. We have to tap it, just we have to touch the tip of the cover slip. So that what happens, the glycerine, whatever the excess glycerine or 1% methylene or saline is coming outside, that is going to be absorbed. Next, precautions. While performing this experiment, what are the precautions are there? How we have to prevent? So procedure I will explain once again. What is the procedure and how we have to perform? So all the material required is this. Overall, this is the material what we need. What we required is this much of material is required to observe the nucleus in the cheek cell, human cheek cell. So once we evaluate the material or the importance. Toothpick is required. Spatula is required. As I told, spatula, that is ice cream spatula or normal spatula in your home. Normally you can take the dissection spatula. You can take the experimental spatula to collect the sample nearby cheek, inside the cheek. So before performing this experiment, we have to wash our mouth thoroughly. Then only we have to perform the experiment. Glass slide. As I told, what is the purpose of glass slide? We need a slide which is made up of glass. Then we need a cover slip. We have to add a cover slip. Next, watch glass. So like this. The watch glass is like this type of situation. Next, needle. Place in the cover slip as well as to spread the cells uniformly. Blotting paper. Excess of glycerine. The purpose of blotting paper to prevent the excess of spreading of glycerine. Whatever the glycerine is coming outside from the cover slip, to avoid that excess of glycerine, we have to add the cover slip. We have to use the blotting paper. Next, 1% methylene blue. So we have to take the solution of methylene blue. The purpose of using the methylene blue solution, the only one reason why we have to use the methylene blue is to make the color. It acts as an identification of the color and act as a spotting material. After that, we have to add the saline. Saline, why we are adding saline? Saline, it contains the sodium chloride solution. So why we are adding the saline? So that once we add a drop of saline, the cells present in the nucleus, it will go for shrinking. Once the shrinking, the water present in the cell wall, the water present in the cell, it comes outside. Thereby what happens, we can easily identify the nucleus. After that, they are saying that we have to add the glycerine. We need glycerine also. Glycerine is required. We have to place the 4 tips of the cover slip. We have to just put a tip of the glycerine on 4 sides of the cover slip. When we do this, what happens? The glycerine, it will make the cover slip not to disturb, not to move from one place to other place, top or bottom like that, it will not do. Then after this, what we need? We need finally a microscope. A microscope is required to observe all this material. So we have observed the material required for the study of the nucleus. In the tomorrow class, we are going to study what is the procedure, how to perform this experiment to observe the cells present in the human cheek cell, especially in the nucleus.